Good afternoon, everyone, and thank you for joining us today. I'm Wendy Heinzelman. I'm the Dean of the Hagem School of Engineering and Applied Sciences and also a professor in the Departments of Electrical and Computer Engineering and Computer Science. And I am thrilled to see that we have such a wide range of registrants joining us today for this session, including we have alumni, we have parents, we have students, we have prospective students, faculty, staff, and medical professionals, all tuning in to hear what Professor Doug Kelly is going to talk about today. So in order to introduce the event today and the speaker, I want to talk a little bit about the work that Doug Kelly and his team are doing. So they work on a fluid that surrounds and permeates the brain and plays a crucial role in brain health. This fluid flows through the tissue, sweeping away waste and delivering nutrients as it circulates around the brain. And this process primarily occurs during sleep and helps to clear proteins linked to diseases such as Alzheimer's and Parkinson's. So at the University of Rochester, a team of engineers collaborating with neuroscientists and doctors is uncovering the mechanisms behind these brain fluid flows. And their research is driving innovations to modulate these flows, aiming to help improve our brain health. So today, Professor Doug Kelly is going to talk about the groundbreaking research that he and his team are doing. They're going to highlight some new insights into swelling during strokes, a potential technique for targeted drug delivery, and promising experiments that they are doing using existing drugs to enhance brain fluid circulation even as we age. Now, before I introduce our speaker today, I would like to offer my deep appreciation to University of Rochester alum Bob Huang and his wife Lily, who had the foresight to make today's program possible through a gift that supports programming and technology and innovation. Today is the 5th virtual event for the Huang Speaker Series in Technology and Innovation. So a huge thank you to Bob and Lily for their support of our university. So today we are thrilled to have Professor Doug Kelly, who is a professor of mechanical engineering at the University of Rochester, as well as a senior scientist at the Laboratory for Laser Energetics. Doug is going to talk to us about cleaning up the aging brain. Thank you, Doug, for joining us. I'll now turn it over to you.
Thanks, Dean Heinzelman. Thanks, everybody, for coming out today to hear a bit about our work. I'm excited to get to tell you about it. Cleaning Up the Aging Brain is my title. And as Dean Heinzelman said, this is highly interdisciplinary and collaborative work. I don't think it would be possible for any one group to do these things singly. And so I want to start by thanking my collaborators. Megan Nedergaard is the neuroscientist here at University of Rochester who pulled me into this topic. And her team produced all of the experimental data you're going to see. Ting Du is a faculty member who works closely with Megan, and actually her hands did many of the experiments you'll see. And Jack Thomas is a senior faculty member in mechanical engineering with a lifetime of fluid dynamics expertise. He's a great mentor and collaborator to me. So my story starts with the big idea that there are two circulation systems in the brain. One is the blood, highlighted in red here in this MRI image of a mouse's head. And the other is the water-like fluid that, as Dean Heinzelman said, surrounds and permeates the brain. We call it cerebrospinal fluid, or when it penetrates in between cells, we call it interstitial fluid. It's all very nearly water. And it not only protects the brain mechanically and keeps it floating there, but it flows. And by flowing and circulating, it can transport wastes and nutrients and anything else around. One way to see that is to do what was done to produce this movie that's playing now. Inject a bit of dye into the back of the skull of a live mouse and watch what happens. And if you do that, you see that the dye spreads quickly around the skull and largely follows the paths of the big arteries around the brain, even though the injection was not into an artery, it was just into the cerebrospinal fluid in the skull. That was an experiment done in a mouse. People have done similar ones in rats and rabbits and pigs and even humans, seeing broadly similar results. So this circulation system seems to be common in brains of many different organisms. Now, the fact that there are two circulation systems in the brain, in a way, is not a surprise because there are two circulation systems in the rest of the body. In addition to the blood, there's the lymph system, sketched here, and composed of a separate set of vessels from the blood vessels. And the lymph system picks up fluid that has seeped out of the blood and trickled through the tissue and returns it to the blood and back to the kidneys and so forth. And in doing so, it also picks up any cellular wastes that may have been seeped into the fluid as it was trickling through tissue, serves as a route for immune surveillance and for immune cells to move around as well. So everywhere else in the body, there are two circulation systems, and there are in the brain as well. That has important implications for human health. So as Dean Heinzelman said, healthy circulation of cerebrospinal fluid in the brain can sweep away metabolic wastes, which our brains make every day normally, but whose buildup correlates with diseases like Parkinson's and Alzheimer's. This circulation can also go awry in pathological cases like stroke, which I'm going to talk about in a minute, or traumatic brain injury, and cause terrible swelling that damages tissue. So there are very practical reasons to study this circulation system. Let me tell you a little more about it. Okay, so I said that in a way it's not a surprise to have two circulation systems in the brain because there's two everywhere else. But in a way it is a surprise because there are no lymph vessels in the brain. They come as far as the edge of the skull, as shown here. And actually, that was only recently rediscovered, but none penetrate the brain itself. And that's long been a puzzle to science because it's well known that the brain is very metabolically active, uses lots of energy. Actually, your brain is 2% of your mass, but uses 20% of your glucose. So it seems like it would need a circulation system. But there's no lymph vessels in it. Okay, so what we're coming to understand is that it does have a circulation system for water-like fluids, but they just don't, it doesn't use lymph vessels. There are different pathways. And a big role is played by what are called perivascular spaces. So peri means around, and vascular means having to do with blood vessels. Most blood vessels in the brain are surrounded by an open space, and that's not common elsewhere in the body, but in the brain it is. And that open space is filled with water-like fluid. And the big idea of this circulation system, which is called the glymphatic model, and was given that name by Micah Nedergaard and her collaborators in 2012, is that cerebrospinal fluid enters the brain tissue along the perivascular spaces that surround arteries, trickles through spaces in between cells, and then comes back out again to complete the circulation along the perivascular spaces that surround veins or nerves. And from there, it gets picked up by lymph vessels at the edge of the skull or trickles out through the spinal cord. Eventually, it's picked up by the lymph system, goes back to the blood, the kidneys, the liver, and so forth. And as Dean Heinzelman said, this circulation system is mostly active during sleep. So here are images of the central nervous systems of two different mice. Both of them had dye injected at the back of the skull. And these images were made half an hour later. The animal on the left was asleep for that half hour, and the animal on the right was awake for that half hour. And you can see that there's much more dye in the brain of the animal that was asleep because there was more circulation going through the brain. Whereas in the awake animal, there's less dye in the brain and much more in the lymph nodes. And we believe that happened because the circulation wasn't going through the brain much. It was just getting shunted out to the rest of the body. That's consistent with observations in a paper published in 2013 by Mikan's team, where they looked with much higher magnification at a much smaller part of the brain and basically found the same results, that dye injected to the back of the skull penetrated much more quickly and much more deeply into the tissue in sleeping animals than animals that were awake. Okay, so we want to know more about how this works and how it matters for human health and how the knowledge of this circulation system might be used to improve human health. And to get there, well, we want data. And so we point microscopes and learn as much as we can from that. Here's an example from perivascular space on the surface of the brain. You're looking at an artery atop the brain of a live mouse. You can see the red blood cells rushing along. Around that artery is cerebrospinal fluid, this water-like fluid. That's what we're really studying. It's in a perivascular space. It's dyed blue here. And we've also, in this experiment, injected some 1 micron fluorescent plastic tracer particles. They show up green in this movie. And we injected them so that we could see the motion of the fluid. They get swept along as it flows. And you can just watch it happen. And the contribution of the engineers to this movie is we used custom written image processing code that finds all the bright green dots in each frame of the movie and then connects dots from frame to frame to frame so that we get a measurement of the position and velocity of each dot in each frame of the movie. And that turns out to be tens or hundreds of thousands of measurements for a half-hour experiment like this. So we can get a lot of data. And we can do lots of things with that data. For starters, we can draw on the colorful comet tails that appear in this movie that indicate where each particle has come and how fast it was going as it came along. Another thing we can do is average this kind of measurements over time to get the mean speed. Here's an example of mean speed in a different surface perivascular space. And we see speeds on the order of 20 microns a second on average, sometimes up to 100 microns per second or so. We see that the flow is fastest in the narrow regions of the perivascular space, which makes sense because if you want to get the same amount of fluid through a narrower passageway, you have to push it faster. We also see that it's slow in the inside of the bifurcation there, which is interesting because it's known that waste builds up there. And we can tell from the data in a very robust and quantitative way what you can probably see by eye. The flow has a net direction and it also pulses faster, slower, faster, slower. And that pulsation happens in synchrony with the pulsation of the artery wall. You get a pulsation every time the heart beats, and you also get bigger, slower pulsations associated with changes in brain activity. Those we have learned are really important for pushing this fluid around. Okay, so we use collaborative methods like this and data mined out of experiments and image processing like this to go after a variety of scientific questions about health and aging. I'm going to tell you 3 short stories about them. And the first is about stroke. So an ischemic stroke occurs when a blood vessel gets blocked in the brain, and therefore part of the brain gets starved of blood. And without blood, the neurons are not getting the energy they need. They're not getting the oxygen they need. They can't keep doing their jobs. And a big job that they can't keep doing is running their sodium and potassium pumps to maintain an ion gradient so they can fire. And so without energy, neurons, they finally give up and they just dump all their ions into solution. And that overwhelms their neighbors, who then dump all their ions into solution, and so on. And it's well known, it's in textbooks about stroke, that this causes what's called spreading depolarization. All the ions go awry, one after the other, spreading across the brain. All the neurons go awry, one after the other, spreading across the brain. And textbooks will tell you that after that happens, fluid comes rushing into the cells and causes terrible swelling. And swelling of brain tissue is really bad. Swelling of brain tissue causes major damage, often irreparable. This image I'm showing is the brain of a mouse that was subjected to stroke. And I am not a medical doctor, but even I can see that this white region at top left is not healthy, and probably most of that tissue never would have recovered from this stroke. So that's the textbook story of stroke. And more than that, the textbooks say that this fluid that rushes into the cells comes from the blood. But we've been thinking about this other circulation system of cerebrospinal fluid and wondered whether it might play a role as well. And so Ting Du did experiments and developed this method to induce stroke by blocking a vessel, and watch what happened. So I'm going to show you two movies at once. It's actually simultaneous recordings in two different colors of the same mouse brain. At top in green, you're going to see neuronal activity. So this is a genetically modified mouse whose brain literally glows in the dark when the neurons are busy. And then at bottom, you're going to see the dye that Ting injected at the back of the skull, which indicates how the cerebrospinal fluid is flowing. And let me start the movie. Okay, so normal neural activity, we get just sort of pale green like this. And in a moment, the bead will get injected at time 0. And you see some normal amounts of inflow of dye at the bottom. But very soon, you'll see this bright green wave at top. That's the spreading depolarization, where neuron after neuron is going awry. And following that bright green wave is a really big, fast inrush of cerebrospinal fluid. The movie's looping. It'll come back around in just a minute. If you watch the green wave last time, watch the dye this time, right? About 6.5 minutes. Okay, maybe 8. There it is. There's the green wave, and look at the dye below just rushing in. It's a much faster flow just by eye than occurs normally. And it only occurs in the region of the stroke. We used image processing to quantify how long it takes for that spreading depolarization to arrive at different parts of the brain. We used some more image processing doing the same thing with the dye, and we were able to quantify in several animals in a statistical way that the fluid always does rush in after the spreading depolarization and always follows it. We were able to quantify with more image processing code how quickly the fluid flowed in. It accelerates after the spreading depolarization passes. So the two seem to really be linked. And we wondered then, how does it work? What's the mechanism, right? I'm an engineer. And so part of my job is to sit in the corner and raise my hand all the time and go, okay, how does it work? We wanted to know how it worked. And to understand that, we zoomed in more with a different microscope and looked quite closely at just one blood vessel and one perivascular space. And that's what I'm showing you here. And when the movie loops, okay, notice right there at the beginning, the vessel shrinks quite rapidly. And that is synchronized with the passage of the spreading depolarization. So when the neurons give up, it messes with the blood vessels and they shrink suddenly. And then the surrounding space was left much bigger than it was before. And so fluid comes rushing. And you can see this really huge, fast inrush. There it is, of tracer particles and cerebrospinal fluid. And that fluid goes on to enter cells and cause swelling. And it happens over and over again in multiple animals when we do similar experiments. It's statistically significant. So this spreading depolarization constricts the arteries and draws the cerebrospinal fluid into the brain tissue to cause swelling. We published that in Science in 2020, and it has potentially important implications for treating stroke because, again, all the textbooks say the swelling comes from blood. But what we found is that's not true in the first half hour. Later, you get more swelling from blood. But for the first half hour, the swelling comes from this big inrush of cerebrospinal fluid in the glymphatic system. And that means for the first half hour, a stroke victim should probably be getting different drugs, and drugs in particular that reduce the circulation of cerebrospinal fluid in order to minimize swelling. Okay, that's one story. Here's my next story. It's about drug delivery. So the brain is a very special place in the body, and it's treated as such. One way that it's treated as such is it's encompassed by the blood-brain barrier. So there's a much more selective barrier between blood vessels and brain tissue than around the blood vessels elsewhere in the body. And little stuff like oxygen and glucose can get back and forth to the brain cells really easily from the blood, but larger molecules mostly get blocked. And that's good for brains because it keeps the viruses out and it keeps the bacteria out and it keeps toxins out. But it can be bad for medicine because it also tends to keep drugs out. And if you inject a drug, into somebody's arm that's supposed to help with something in their brain, probably only 1% or 0.1% of what you inject is ever going to get to their brain because of the blood-brain barrier. One idea to improve upon that situation is to use not the blood circulation system, but the glymphatic circulation system to deliver drugs to the brain. And that's already done for epidurals during labor, for example, where an injection is given right into the spinal cord and makes its way up to the brain. We think there's potential to do more things like that, and we wondered about how to make it work. And Megan had a great idea that you might be able to promote circulation of cerebrospinal fluid when you want to bring a drug in if you think about osmosis. So, okay, when you sit in the bathtub for a long time, your fingers get all wrinkly, right? That's osmosis. What we did was we, again, injected some dye into the back of a mouse's skull so that we could see what was happening with the circulation system. And we injected the mouse with a high concentration solution of mannitol, which is just a sugar. It's just very sugary sugar water. And that sugar water was meant to do to the mouse's brain cells what the bathtub does to your fingers, that is, make the brain cells shrivel up a little bit by sucking water out of the brain cells. Once they're shriveled up, there should be more space in between them where circulation could occur. So that's the experiments that we did, and that is what we saw. Here are images of dye concentration in a mouse's brain over time, kind of like that movie I played you in the beginning. And the top row is just a normal animal, and the bottom row is an animal that had this sugar water injection. And sure enough, you can see that dye penetrates more quickly and more deeply into brain tissue with the sugar water than without. And we did image processing to quantify that. We went a little further than that even. My team had previously, for a very different application, invented what we called front tracking, where we wrote code to do image processing to find the leading edge of a bright or dark region in a movie and track it over time and measure how fast it moves. And we did that with the dye injected into these mouse brains. And we're able to show that, sure enough, when you inject some high concentration fluid to shrivel up the brain cells, you get faster flow, and we could see where it was faster as well. This, by the way, is salty water instead of sugary water. We did both, and they both work. And we published that back in 2018. And we think this could be promising for drug delivery. If you inject some salty or sugary water, you can promote this lymphatic circulation in order to sweep in not just dye, but drugs if you need. Okay, my third story for the day, and my last story, is about aging. And I should give you a little background first. It turns out that aging really inhibits this lymphatic circulation in the brain. And Mike and Edegard's team did some experiments demonstrating that quite convincingly that they published in 2014. One thing they did was to inject some amyloid beta deep into the brain, as sketched here, again, working with mice. And they did it in young mice, middle-aged mice, and old mice. And then an hour later, they measured how much of what they injected had been swept out of the brain. And they found that much more got swept out in young brains than in old brains. Amyloid beta, by the way, is the protein whose buildup correlates with Alzheimer's disease. So they did this with some other dye and such as well, but they did it with that protein in particular because they were thinking about Alzheimer's. Another experiment they did was to inject some dye at the back of the skull again and see how quickly it was swept into the brain. And usually we're worried about waste being swept out, but it has to be a circulation. If fluid's coming out one place, it's got to be coming back in somewhere else. And so, sure enough, in young brains, that dye injected just in the skull got swept into the brain much more quickly than in old brains, again showing there seems to be less circulation with age. They went on to look at mechanisms that might cause that circulation, and one they looked at was the pulsation of blood vessels. I remember I mentioned earlier that these flows in the perivascular spaces go in sync with the pulsation of the blood vessels. We think pulsation of blood vessels is a major driver of these flows. There's lots of evidence to show that. And they found that in old mice, the penetrating arteries in particular, so not the ones that lay on top of the brain, but the ones that dive into brain tissue, pulse a lot less than in young mice. There are also fewer nearby water channels, it turns out. That's important, but we'll talk more about it today. So, okay, aging inhibits brain circulation, and one of the mechanisms that causes that is less pulsation of blood vessels in the brain. Now we know that ultimately this circulation through the brain gets picked up by lymph vessels. At least a lot of the fluid gets picked up by lymph vessels. I showed you some in the skull earlier. So more recently, we wondered whether that too might be degrading with age. And so we went to study. We pointed a 2-photon microscope at some lymph vessels, and by we I mean Ting-Du, and injected microspheres again at the back of the skull, along with some dye just to make the lymph vessels easier to see and find. And here's an example of the results. You can see the vessel pulsing. You can see particles being swept back and forth, and we have path lines drawn on because we're tracking them with our code. And at right, you can see we're also using automated image processing code to track the diameter of the vessel because lymph vessels pulse a lot. And in particular, so this is a cervical lymph vessel. It's in the neck of a mouse. It's muscular and it contracts and expands almost like a little heart. It's almost like a string of little hearts because it contracts quite a lot in normal healthy animals, as you can see here. And there are valves every so often to ensure that the flow goes in the right direction. So we wanted to track its contractions. We've got the diameter plotted at top right. We also were tracking particles and we wanted to see how fast they were going. So that's the downstream velocity at right in yellow. And meanwhile, we were measuring the heart rate, that's ECG in red, and the respiration rate in green. Okay, so once we could measure all this and start gathering lots of data, well, we looked at mice of different ages. Here's a 2-month-old mouse that's pretty young, an 18-month-old mouse that's pretty old, and a 22-month-old mouse, which is really old. And you can see that the diameter of the vessel of the young mouse at left pulses very regularly and a lot. There's much less pulsation of the 22-month-old vessel at right. You can also see that the flow speeds, the downstream velocity in the young mouse's vessel is much faster than in the 22-month-old at right. So yes, by eye, it is the case that age reduces flows in the lymph vessels that are draining waste-laden fluid out of the skull. From movies like that, with our automated code, we were able to extract lots of data, and we can plot how diameter varies over time and show in robust quantitative ways that lymph vessels pulse less frequently in old mice. And we can show that the flow in the vessels is slower in old mice. And we can show in particular that there's more backflow. You could see when you were watching the movie that the fluid in the lymph vessel would rush forward for a while and then turn around a bit and then rush forward again. There's much more backflow in the vessels of old animals than young ones. So thinking mechanistically again, we wondered about that. Looked a little closer here. Oh, there it went. Here are 2 valves in lymph vessels for a 2-month-old mouse and a 22-month-old mouse. When the movie loops, you'll see at left the valve on the young mouse actually flops open as the flow starts, which is how they work. On the other hand, the valve in the old mouse really doesn't flop open and close much. Let's see, here comes the movie. Watch the young mouse. Where'd it go? Oh, here it is, here it is. Yeah, there it goes. Okay, opened up. So we saw that the valves weren't really doing anything. We wondered whether the valves themselves were too stiff or whether the flow through them was too weak. We wondered why weren't the valves opening and closing as they should be. And we looked real closely at the cells in the vessels and stained the tissue with particular dyes that only go to smooth muscles and particular dyes that only go to cell nuclei. And what we found looking at where the smooth muscles were is that the vessels aren't covered by muscle nearly as well in old mice as in young mice. But counting those blue nuclei, we learned that there were no fewer cells in the old mice. So it wasn't that a bunch of muscle cells had died and disappeared. They'd just gotten old and didn't cover as well and didn't seem to pump as well. So then we wondered, since the muscle cells are still there, could we stimulate them? To do that, Ting and Megan had the great idea to use a drug called prostaglandin 2 alpha. It's known to stimulate smooth muscles. It's often given to induce labor in humans, actually. It's approved for human use. And injecting some of that, sure enough, we saw that flows in the lymph vessels of old mice sped back up to speeds similar to what we see in young mice. The pulsation was restored. Yeah, there's the speed, and there was a lot less backflow. And this work we published just last fall in Nature Aging. And we wonder if there are clinical implications for this too. This drug, again, has never been used for this before, but is approved for other uses in humans and there is therefore presumably fairly safe and might be used for things like this in the future. That's still a few years out. You know, we've only done mice. There would have to be clinical trials and such, but we wonder and hope if this might help people maintain good circulation in their brains as they age. Okay, I want to conclude by thanking some more folks. This is my research team. They make it fun to come to work every day. These folks range from a research assistant professor to some staff researchers, postdoc, a bunch of graduate students, some undergrad researchers, so sort of folks at all ranks. And among these folks are the people who did the heavy lifting and the data analysis I showed you today. I also want to thank the agencies that fund our work. None of what you saw today could have been done without research grants. All of these people you see are paid researchers, most of them full-time, and they're all paid directly out of grants that my collaborators and I have won from sources outside University of Rochester. The Army Research Lab has funded us because they know that these circulations happen during sleep, and soldiers are sleep deprived all the time. So the Army wants to know, how does this thing work? And can they do something to help sleep deprived soldiers be smart and maintain their health? We've received a few grants from the National Institutes of Health, one all about how the mechanisms of these flows vary between wake and sleep, and actually different stages of sleep, like rapid eye movement, non-rapid eye movement. Another is all about just directly, how does all this tie to Alzheimer's disease? So that's really essential. And, you know, as maybe you've heard, there's lots of uncertainty right now about federal research funding in America. There's lots of proposals for cuts. If it weren't for the funding, we couldn't be doing any of this. And I think it's valuable. I'm passionate about it. So of course I think it's valuable, but these findings have direct implications for human health. The Army thinks they're important for national defense. There are tech transfer efforts coming out of this. These people get to work for a living. There could be patents. There could be economic activity out of this. We think that research really adds a lot to our country and its prosperity and security. If you think so too, please support research however you can and consider speaking with your congressional representatives. Okay, with that, I will stop. Thanks very much for your time.
Right. Thank you so much. That was just absolutely fascinating. And we have a lot of questions coming in. I just wanted to let people know that you can type your questions into the Q&A, and we'll try and get to as many of these as we can. I wanted to start with one for you, Doug, about for those listening today, during their awake hours, during the daytime, is there anything they can be doing? What can we all do to ensure that our glymphatic system has the best opportunity to function when we're actually asleep?
Yeah, exercise is really good. Being active, being socially active, engaging in like intellectually challenging things in your life, those all really help. I saw a research paper just a couple of months ago that was a review of a huge amount of prior papers about Alzheimer's disease and risks for Alzheimer's disease. And the conclusion was that about half the risk just comes down to lifestyle, comes down to how well do you eat, how well do you take care of yourself, don't drink too much, don't smoke, etc. So you have a fair amount of control, and most of it is live a healthy lifestyle.
Great. Something that I'm very interested in, and I think probably many people out there are, you're a mechanical engineer.
Yes.
This is very, very interdisciplinary work, and curious both how you got involved and how others, especially students, can get involved in work that really sits at the boundary of multiple disciplines.
I love telling this origin story. Micah Nittergaard pulled us into this. The way it happened was she was invited to apply for a grant in late 2016, early 2017, and wanted to have some fluid dynamics expertise on the team. And I think she started looking in the university catalogs to see who taught fluid dynamics, and she found Jack Thomas first. And he said, "Hey, we need to pull in Doug and some other people." And so we sat down to have a meeting. I had read that 2013 paper, which is just an amazing paper, but I didn't know anything else about the work beyond that. And in the second meeting, Megan's PhD students showed data not as nice as this movie that's here, but along these lines, that they could image and make movies of tracer particles inside the brains of live mice. And I had no idea that was possible, but I'd come out of fluid dynamics and turbulence experiments. And for almost a decade by then, I'd already been using image processing to make measurements based on movies of particles. And nobody had ever done it in the brain before, but right away, I knew what to do with it. It was a wonderful serendipity, and the thing just kind of snowballed from there. You asked about how people can get involved. I think, well, we're always looking for good people is one answer.
Great. Curious about, you talked about using, I think it was either sugar water or salt water to basically shrivel up some of the cells in the brain to provide more space for the fluid to flow, which is fascinating. Two questions that have come in about this. One, isn't that dangerous for the brain? Like, does it hurt the brain to have the cells shrivel up in any way? And then the second is, this sounds like a really great opportunity for drug delivery. Where do things stand? Is this being further developed? Are drug companies starting to think about using the glymphatic system as a way to deliver their drugs more effectively?
Okay, it doesn't cause so much shrinkage to cause damage. Maybe this might give you a headache. This would probably give you a headache. But cells grow and shrink all the time naturally. In fact, one of the findings in that 2013 paper was cells in the brain shrivel and then expand again as we go to sleep and wake up. This is probably one of the main reasons that these circulations happen more during sleep, because the cells shrivel up a little during sleep and leave more space. Let's see, where does it stand? I believe they filed patents on that. There are definitely lots of people working on drug delivery. I know folks in startup companies, and for that matter, folks in quite large biotech companies looking at how can you better get drug up from a lumbar injection. And they're thinking about, all kinds of ways to modulate these flows to get drugs in.
Great, great. A couple of questions have come in about the stage of sleep. Does this happen at all stages of sleep? Is there a particular stage of sleep where this happens? And how does that work? And then along those lines, another question that has come in is, does it make sense? Is it helpful to get more sleep, or is there sort of, your typical 8 hours and you're good?
I think 8 hours and you're good is probably right. These flows happen most of all during NREM sleep, the non-rapid eye movement sleep, which is the deepest sleep. And, we wonder about whether consciousness and these kind of flows are incompatible somehow. We don't know for sure.
Mm-hmm.
It's also true that microarousals are common during sleep, where your brain sort of wakes up for 10 seconds or 20 and then conks back out again. You're usually not even conscious of them. And in mice, at least, microarousals really push a lot of fluid around. And so having a lot of microarousals during sleep causes a lot of circulation.
Which would obviously help in the end. And so
Yes.
making sure, but I think this is probably a good indication of why sleep is so important. I mean, we've known for years, right, that getting 8 hours of sleep, especially in kids, is really important. And this is just another example of why that is the case. Where does the fluid go when you're removing all the waste? Sorry, not the fluid, but where does the waste end up? Is it just how does the body get rid of all that waste?
Sure. Ultimately, it's going to get picked up by the lymph system and put in the bloodstream and filtered and broken down and recycled in the kidneys and liver.
Okay. So it becomes part of the regular waste cycle of
Right.
the human body. A question came in about microplastics. I don't know if you know the answer to this, but it's an interesting question because I know there's a lot of concern about microplastics and how they're getting into the body. And could those potentially be getting into the brain and then reducing the area or the flow, the volume where the flow is happening? And could that potentially be causing some of these issues that we're seeing?
That's a really interesting question. I haven't seen anybody get as far as studying that in detail. There, I saw a journalistic piece in Science a week or two ago about a researcher who was finding lots of microplastics in brains. It gives you pause. It's a little scary. For sure, some of the passageways are quite tiny. The ones I've showed you are much too big to be blocked by microplastics.
Okay.
But when the fluid starts trickling through between cells, those are tiny. That some of those places could get blocked. There's a lot of passageways, though. Maybe it doesn't cause too much trouble. That's a great question for research. I hope somebody will do that soon.
Yep. Along these lines, there's a question about, are there companies or startups that are kind of taking this work and running with it?
I haven't been directly involved with any startups.
So
But
U of R does not have any startup companies that have come out of this work.
it gets real close to, no, the short answer is no. U of R doesn't have any startups that come out of this work.
Okay, great. Let's see. Other questions are about, so you talked about the way that you kind of got into this area. And it seems like you've learned a lot about the biology and the medicine of how things flow through the brain with the lymphatic system. I would imagine that your collaborators over in the Med Center have learned a lot also about engineering. Can you speak a little bit about sort of the benefits that you see to having people with lots of different backgrounds coming together to think about these problems? Like, have you been able to, do you think in your mind, has that interdisciplinary collaboration led to new discoveries that wouldn't have been possible without that?
Absolutely. Absolutely. It has. I mean, I told the origin story, and particle tracking was a relatively routine method already in 2017 in the fluid dynamics community. But apparently we were the first people to cross paths with the neuroscientists who had this kind of data, because nobody had done automated particle tracking before we did. And actually, hardly anybody does it even now, even though we give our code away, which is kind of a mystery to me. But anyway, I've always felt that the best advances happen at the boundaries between fields. And yeah, we've learned a lot from each other. I think that the neuroscientists and the engineers talking closely and working closely together, because, well, the engineers, we stand in the corner and say, how does it work? What's the mechanism?
Right.
And we're often trained to think about counterfactuals and think about simplified models, which don't at all capture all the biology and therefore aren't perfect, but can give quick insight into like particular mechanisms and how to control things. I think that's a helpful way to talk about it. And we've been able to produce way more numerical quantitative data with all the image processing, which I think is an important contribution. And I should say too, I've drawn paychecks from 8 universities over since I was an undergrad, but I've never seen one where the boundaries between departments are as low as they are at University of Rochester. I think that's a great strength of this place. I really like it.
Absolutely. A couple of questions about drugs. Sometimes people use drugs to help them sleep. Does that mess with this lymphatic system and the cleanup of the brain? And then the second question is, have you looked at existing medicines, existing drugs that can help things like with the muscular, with the rigidity of the vessels that you were talking about? There's some existing drugs that can deal with those. Have you thought about or looked at how you can use existing approved drugs in these new ways?
We haven't published anything about manipulating the vessel stiffness with drugs, except in that 2018 paper early on, we induced high blood pressure in mice, which the vessels are programmed, if you will, to keep the same size. And so if you increase the blood pressure, all the cells have to flex harder to keep from swelling out. And then that changes the stiffness of the blood vessel and changes, it actually reduce the amount of lymphatic circulation. And so that's one of the big pieces of evidence we have that pulsation pumps the cerebrospinal fluid around. For sleep drugs, I think I haven't seen a real careful analysis of that. We have done, Lauren Hablitz over at the Medical Center led a really good study of different anesthesias used in experiments with mice in the lab. And I tried 6 different ones, and some really allowed these flows and some really damped them. So it certainly could matter. I would first say that any sleep aid that a person takes that makes them sleep but prevents good, deep NREM sleep probably isn't the best for circulation in the brain.
Great. And I think we're coming up on our last question, but I wanted to ask a little bit about, do you work with, outside of the clinical physicians, have you talked or worked with anyone in psychology or psychologists, psychiatrists, to think about things with mental health and how this could potentially impact mental health in a way similar to how it's impacting with Alzheimer's and Parkinson's?
I have not gone that far. My collaborators have certainly done studies looking at behavioral changes in mice, but I haven't gone as far as human psychology. We have a collaborator at MIT, Laura Lewis, who does MRI scans on people and, does stuff like play sounds or flashlights, or have them do activities while they're getting scanned to see how it changes the flows. I don't think she's got as far either as long-term mental health, however.
Interesting. Well, thank you so much. This was, I think we all learned a lot about our brains, about aging, about what we can do, and also just about the importance, as I'll just reiterate what you said, the importance of research and how we need to keep funding our research. We need to make sure that we are able to have these sorts of discoveries. They help us live longer, happier, healthier lives. Really, really important. So thank you for all the work you're doing. Thank you for your team, for all of the contributions that you all are making. And I just want to say a big thank you to all of those of you who are joining us today on the webinar. We hope you enjoyed the program. A recording of this program will be sent within the next week or so, along with some follow-up articles. So if anyone has questions we didn't get to, we'll send information that has more ways that you can find out about the glymphatic system, about the work being done by Doug Kelly and his collaborators. With that, I hope everyone has a wonderful day and hope to see you at another event for the University of Rochester real soon. Thank you.